Hello, my name is Angela Columbano and I am a Director of Business Development and Partnership at Genethon. It's a pleasure for me to be here today. We're going to spend the next few minutes going through a presentation about Genethon, our programs, and our technology. So Genethon was funded by the AFM Teleton, which is the French Muscular Dystrophy Association, Patient Association. The AFM Teleton was originated in 1958 by Madame de Queper, who had 4 children with the Duchenne muscular dystrophy. And the AFM has invested over €1 billion since the creation in research and in projects that aim to find a therapeutic solution for rare diseases. So this is really a snapshot of what Genethon is. So Genethon is, first of all, a non-profit research and development organization. We have an integrated R&D center based in the south of Paris that ensure translational development from research from the bench to the bed to the clinical development. And we have been created in 1990, and already in 1992 we were responsible for the first publication of the first map of human genome. In 1997 we were already starting working on research of gene therapy. In 2006 we started our own internal biomanufacturing. We have been going through a large number of partnerships with industry leaders, and there are a number of products already on the market and or close to be on the market. The technology that was developed by Genethon or with Genethon, such as Organ SMA or Lumevoq. Today we have 8 products in clinical trials that are either carried out by Genethon or by our partners. There's 10 programs that are on track to be in the clinic within the next 3 years. And we have a total of 200, just over 200 people that are working with Genethon today. So Genethon has been able to create a very large and strong network of expertise in gene therapy, both in discovery and research and innovation, which have translated in over 700 patents coming from the work initiated at Genethon within this network of collaborations. Same as happened with clinical development, where we have been working with the leading clinical centers for gene therapy. And finally, within manufacturing, which is probably one of the most important bottlenecks today, that to allow gene therapy to be available to more and more patients and more therapeutic areas. We have spun off from Genethon a GMP manufacturing facility that today is a CDMO. It was case in one of the leading CDMO for gene therapy in Europe. So Genethon at glance, we have a research team of about 90 scientists. We have a unit of INSERM that is the equivalent in France of the NIH that is embedded within the research team at Genethon. We have our own CMC development with about 40 scientists that are working on breakthrough technology for production, purification, and control. And we have finally a team of experts in clinical and regulatory to bring the product to the clinic. So these are the 4 key. We have the muscle, which is an important area for Genethon. 2 scientific leaders, Dr. Anna Bushbell and Dr. Isabelle Richard, who work on different diseases in neuromuscular field. We have a group led by Dr. Angali that is specialized in immune and blood disorders. Dr. Giuseppe Ronziti that works within the liver and metabolic disease area. And finally, we have a platform of ex vivo hematopoietic stem cell with gene editing led by Dr. Maya Mendola. So this is our pipeline in neuromuscular disease. We have an asset, an AV8-based gene therapy that we developed for myotubular myopathy that is today being developed by Audentes. We have also an AV8-based microdystrophin approach for Duchenne that we are co-developing with Sarepta. We have a number of different programs in limb-girdle muscular dystrophies, which are today part of a spin-off that we are structuring, and they will be soon part of a new company coming from Genethon called Atamium. And we have additional projects that are in preclinical or discovery research. So an interesting program in SMA, PME, and Farber, which are acid ceramidase deficiency with very interesting preclinical results. We have additional approaches in Duchenne, an AAV-based dual vector approach and an exon skipping approach. And we also have a work ongoing on myotonic dystrophy type 1 based on an AAV CRISPR gene editing approach. So this is our portfolio in liver and metabolism. The most advanced program is in Krieger and Najjar, and this is today in the clinic with very interesting preliminary results that I'm going to share with you in a minute. We have an asset in Pompe that's been developed today by Spark, and is also started the clinic this year, phase 1. And then we have several assets, several programs in preclinical, GSD3 and GSD1A and XLH. So Krieger, GSD3, GSD1A, and XLH are part of a second project that we are structuring today for a spin-off called Flamingo. So just a glance of what we are doing in Krieger Najjar. Krieger Najjar is an ultra-rare disease, probably around 400 to 1,000 patients worldwide suffering from this disease. Is caused by the mutation of the gene UGT1A1, which translates in high level of unconjugated bilirubin, and if not treated, the consequences are serious neurological damages and lethality. So today, the only way out of this disease is either liver transplantation, or otherwise the patient have to undergo a long session, about half of the day each day of phototherapy, which is obviously very complex in terms of the quality of life of these patients. And they remain a constant risk of having life-threatening spikes of bilirubin. So we have developed our own AAV gene therapy approach for Krieger. It is an ideal disease for gene therapy. You only need to restore 5% of the activity of the enzyme to have an important impact in terms of the disease. Obviously, the endpoint is the measurement of the bilirubin. So this is a very easy to assess endpoint. We have our gene therapy approach has a very strong preclinical package. We have data in rats. They show long-term correction of the majority of the animals treated. And the stage of the clinical trials in adults, with 2 doses tested so far. So it's a dose escalating trial. There were 3 doses planned. For the time being, we have tested the first and the second dose. With an efficacy endpoint of interruption of phototherapy at week 16. It's the most important. So these are the results which were published recently at the European liver congress at the end of June. So you will see the dotted line are the measurement of total bilirubin on the 2 patients that were treated with a lower dose. And the straight line are the measurements of total bilirubin in patients that were treated with the second dose. And you see a clear, strong response with the first patient has already reached the primary efficacy endpoint. And so that's, these results are extremely encouraging in terms of the efficacy and safety of this product. So one interesting aspect of this disease is that you can visually see very clearly the consequence of the disease within the color of the skin and the color of the eyes. And you can see this is the patient that was treated, the first patient on the second dose, and you can see how amazing the results are in terms of the difference that it made to her. So just a few words on our technology platform and what we want to achieve. Obviously, the important point that will help us to go to the next generation of gene therapy is to enhance efficacy through tissue-specific capsid and promoters and gene editing approach. The second big pillar is how we can overcome the immune response and allow a second dose and also those in seropositive patients. And the third big pillar is the enhancement of bioproduction. And our ambition is to get 100 times increase in productivity through a number of projects upstream and downstream. So an example of our technology platform with regard to with the objective to enhance the efficacy of gene therapy is our platform on novel capsids. They are designed through rational shuffling and with the objective to increase organ tropism wherever needed, depending on the type of disease we are addressing. Low seroprevalence, so trying to reduce or modulate the immune response, and high manufacturing yields. So we have 10,000 different variants that have been identified, and over 100 has been tested in vivo in biodistribution studies in different tissues, and the results are extremely encouraging. As an example of our strategy to improve or to manage the immune response to gene therapy, we have investigated IDES, which is an enzyme of bacterial origin, endopeptidase, that is able to cleave immunoglobulin, to degrade immunoglobulin. We have tested this approach in non-human primates, and we have had extremely interesting results, especially in non-human primates that were seropositive. So they would have been not eligible to gene therapy. And finally, with regards to our investment in innovation in bioprocess, we are trying to tackle all the way through from upstream to downstream, every step of the way, how we can enhance the current yield and productivity. So through novel producing cell, to novel transfecting agents, innovative plasmids, and novel analytical methods. So we have partnerships, as it's seen throughout the presentation, is an important aspect of Genethon. And so we are actively looking for collaboration on existing or novel gene therapy programs, as well as collaboration for our technology platforms. And one of the models that we have started to structure within the last couple of years is this creation of spin-off when it makes sense from a portfolio point of view. We are working on 2 today. At Tamio, there will be at the forefront of limb-girdle muscular dystrophy therapies in Flamingo, which are gonna tackle novel technologies in metabolic liver and CNS disease. So thank you very much for your attention. And these are my email address in case you want to contact me. Thank you.